Welcome to Moving Medicine Forward, a podcast presented by CTI Clinical Trial and Consulting Services. This podcast is centered on advancements in healthcare and elevating individual stories and their expertise. I'm your host, Jeremy Schran, and together with my co-host, Raina Graham,
hello,
we will take you through a deep dive of CTI, clinical research, the patient journey, and much more. Welcome to a special episode of Moving Medicine Forward. Today we are talking all things cell and gene therapy, just in time for the American Society of Gene and Cell Therapies annual meetings. We will have 4 different segments today. First, we chat with Bret Marshall, Associate Director of Business Development and Client Management at CTI. He discusses CTI's involvement in cardiac cell and gene trials. Next, we discuss chain of custody in cell and gene therapy with 2 of CTI's brightest, Michael Schmidt, Clinical Project Manager 2, and Nicole Sparshott, Clinical Trial Manager. After that, with the power of technology, I go across the ocean to Portugal to discuss feasibility with CTI's very own feasibility manager, Joana Lancastre. And finally, we have my favorite segment. We are talking KRAS with the brilliant Jaclyn Guillory, Director of Clinical Project Management at CTI. So let's dive right in with Bret Marshall and CTI's involvement in cardiac cell and gene trials.
Welcome into the podcast, Bret. Introduce yourself and tell us what you do and give us a little bit of your background.
Yeah, well, it's great to be here, and a little bit about myself. So I am currently in a role as a client manager. So I partner with biopharmaceutical companies that have a novel idea, and we are fortunate to work with some of the leading institutions at centers to get patients treated. I kind of came to that through, a roundabout way. Started my career with a bachelor's in biology, knew I wanted to impact patient care. I volunteered in the hospital, had some exposure as an EMT, and found out that actually, the clinical research side fit my interests best because it's a balance of science, but still having an impact to patients. Started with CTI more than 9 years ago as a research associate, was fortunate to work on the early gene therapy for a retinal disorder, and was really struck by just how transformative that was for patients. So, someone that was losing their eyesight at a very early age, almost looking through a straw as their visual field, and receiving a one-time gene therapy and honestly stopping that disease progression. So that was just amazing to me, both from the biology standpoint as well, as just the functional outcomes for patients. And I just continued that career, worked as a clinical project manager focused on cell therapies. Again, similar concept, really devastating diseases, transformative outcomes, for patients. And I guess transitioned over to sales just to have a broader exposure to companies. Really enjoyed, partnering with some of the leading thought leaders across these institutions, as well as novel companies that have a new concept, new technology from basic research, and trying to take that into a patient for the very first time. So that's exciting. I've launched 7 first-in-human programs. So that's always, an interesting mix of bringing perspectives together and really pushing something into reality. And really glad to be at CTI, company that has had a consistent, mission from Tim and the top down to really impact patient care and have patients being the driving force behind all the decisions that we make.
Could you give the audience an overview of CTI's involvement in cardiac gene and cell therapy trials?
Absolutely. I mean, I can talk about current day, but I think to really start the discussion, have to go back to our founding. As many of us know, these rare diseases, there are no curative treatments. And so transplant for the longest time has been the standard of care. CTI was founded in transplant more than 25 years ago. Really an important step in kind of that disease treatment paradigm, but it's, laden with risks as well with immunosuppression. We're talking a lot of times about pediatric patients, young adolescents, just the organ procurement process, being on a wait list, a lot of anxiety around that. And so I think where the field has evolved with gene therapies, one-time treatment, where they can get, a curative, drug that can drastically change the disease progression, or, importantly, their life as a whole, is something that's really remarkable. We've seen this play out in oncology, from the cell therapy side as kind of a proof of concept. We have some great data out there, more than 10 years in some of these oncology patients that received a cell therapy. And so taking that into other indications. And for CTI, that really manifested in rare disease. As a rare disease, you think of it as just affecting a few patients, but collectively, it's a very large number and percentage of the population that are impacted by these diseases. And so as the biology or basic science matures, we identify new targets, and that creates new therapies. We've seen that play out in a couple of recent approvals. I think what we've heard from the FDA and Peter Marks's recent comments is that they're very open to an accelerated pathway of getting these treatments to patients. And so the future is really bright for patients with a rare disease. And I think what CTI does extremely well is to align ourselves with those thought leaders, those partners. And that goes from individuals doing the basic research, a company that's taking that research, translating that into the clinic for patients, and even the advocacy, partners, someone that is really important in understanding the true patient experience and how we can work with those groups to design the trial around it that is feasible for patients. And we can continue that long-term data that's needed to get an approval. And so I think those are the key parts to CTI's approach that really stand out. It's the 1,000 little things that it takes, to get a patient enrolled, a drug approved, and making sure that all those steps are in place with a very time-efficient, cost-efficient pathway to that approval.
So it sounds like cardiac gene and cell therapy trials are very complex. Can you discuss some of the challenges you face with some of these trials and some of those complexities?
Yeah, absolutely. I think it's kind of twofold. There's the technology challenge. Right now, I think there's a lot of companies looking into better vectors, ones that are more specific to the tissue type, that can be administered in a way that's easier for patients, easier for hospitals to give these. An example of that would be, a systemic delivery where you're giving an IV of this drug versus some of the programs we've started with early on have to do a direct administration, intercoronary, intraventricular. Some of those routes are, a standard procedure for pacemakers and others, but when you try to do that in a pediatric patient, for example, it definitely adds complexity. Many of the centers are specialized, so we have to work closely with our teams to find the right center with patients as well. These are heart failure specialists, largely academic centers, which we have a very long-standing relationship with. And so to go to the right place to find the right patient for this investigational therapy and to follow that patient as they mature in life. They'll go to college, they start a family, but the long-term data that you need for an approval is a key part of that. And I think probably the second part, so that's more of the operations. The second part is the regulatory framework and what the FDA is expecting and how we can generate the data that's mature enough to demonstrate a benefit and to say that this is safe. And I think this is where we've seen the most change recently. It's great to see regulators are open to novel approaches, and it's kind of innovative solutions that we're bringing forward, whether it be in wearable technology to monitor patients in a more convenient way, linking, external data sources through some type of comparator control. CTI, I kind of mentioned, worked in, over 75% of our trials are in rare disease. That has ranged from, studies as small as there being 100 known patients in the world with the disease. How do you run a comparator trial in that? How do you recruit enough patients? And you can't really give them a placebo to control against it. So these are some of the questions that our team are working to solve. We have experience linking up, standard of care data or, maybe a data set and advocacy group, for example, to show what that disease progression looks like and how we can define a surrogate that will tell us earlier, will this therapy work and will it have an impact so that we can get an approval and get this into more patients faster than waiting for some type of ultimate outcome like death or survival. It's kind of a morbid approach, but really, one thing that we've done that I think is really important is to bring in like a model-based approach. And the FDA just did a pilot study where they're using model-informed drug development. One aspect for gene therapy is a disease progression model. And so what that really looks at is what's the natural course of a disease how can we understand what the end kind of disease progression is for these chronic patients and identify biomarkers as a surrogate to demonstrate that if we affect this biomarker, we will impact the ultimate disease. And so it's a pretty novel approach, but one that's really impactful for these small patient populations and long-term data that the FDA requires for an approval.
So you talked about how transplantation, rare disease, cell and gene therapy are ingrained in kind of the history of CTI. You also talked about the challenges and how we approach those. Can you share a success story from you've been involved in a lot of trials around in this therapeutic area. Can you talk to us about a success story in cardiac gene and cell therapy trial?
Yeah, I mean, we've been fortunate to work with a number of companies, kind of the first movers are first to really take that leap and say, we're going to tie ourselves to gene therapy as a therapeutic kind of treatment option. And that's resulted in approvals outside of cardiac. What I'm most fortunate or, excited about for the future is that we are taking kind of this proof of concept and moving it into larger patient populations, specifically for heart failure. It's impacting close to 2% of the world population. And so, how do we take a small kind of niche therapy into larger applications? And CTI is doing this, with a number of companies. I think to your question, what are we most proud of? It's really starting with that end in mind and devising a strategy for our clients to get there as quickly as possible, to de-risk assets using modeling before they ever get to a patient, and then to file or negotiate, certain accelerated approval pathways like prime designation, orphan, breakthrough, things that materially benefit our clients that are developing these products and also patients. And so, from recent experience, we're working on a phase 2 pivotal program that had very similar challenges to what I've discussed earlier. Very small patient population, transplants the standard of care. It's a chronic condition in pediatric and adolescent patients that have kind of different disease severity. So how do you find that right patient and how do you test against the control? And so we are collaborating with various groups to try to improve, the recruitment by spreading awareness on genetic typing, which is a key challenge there, to get those patients into a trial to demonstrate that follow-up data that's needed. And so it's moving forward on a global scale. I think one thing in rare disease, as you have these accelerated pathway approvals, you have less time to prepare for a commercialization. So there's a lot you have to think about upfront and have moving in parallel. And so having kind of a multidisciplinary team that knows what we're trying to accomplish at the end of the day, and then functional or domain expertise in those areas to make sure that all those components go together is key, because you're running, a natural history study in parallel to an interventional trial so that you can basically have a comparator to demonstrate this works. And this should be available to more patients. I think working across, our global footprint gives us an advantage where we can go direct to patients. We can kind of survey the landscape of what certain countries will require, certain investigators want to see for their patients, and make sure that we build that in and understand that there's differences in how assessments that are needed to demonstrate this proof of concept or this approval path can change. A 6-minute walk test is one of those, pretty standard, but there's a lot of interoperator variability. And so how do you design a trial that is consistent across multiple centers, geographies, regions, is something that, is critical, and our team's doing a great job. And we're moving towards, that BLA filing towards a submission and an eventual approval on that instance.
As we're moving things forward, as we're moving medicine forward, if you will, what are we looking forward to, and what are CTI's goals for the future of cardiac gene and cell therapy?
It's a great question. I think try to take it from the lens of what are the current constraints and how we're working to solve those. And one of those is genetic typing. So there's an advocacy group just north of Cincinnati where we're located that's trying to tackle this problem. Less than 1% of patients are genetically typed. Close to 50%, as I recall, of genetic cardiomyopathies, or cardiomyopathies have a genetic component. And so there's a big gap there in awareness. Patients don't know, what therapies are available to them because they're so precise down to the gene until they get that genetic typing. And so we're actually a corporate sponsor for the DCM Foundation, working to spread awareness to these referral centers, the cardiologists, that, genetic typing is important to the disease progression, the patient's care. How do we get those patients then transferred to a specialist center like a Cincinnati Children's or a Christ Hospital that has the familiarity with genetic typing, the ability to deliver gene therapies to patients of all those patients, and how do we make sure that we're supporting those patients in that diagnosis. If you get a genetic diagnosis, there's a component of that that's pretty anxious. You may have the disease, but it's not manifesting itself yet. You may be a carrier and pass that on to family members. And so there's this whole kind of umbrella of support that patients need. And beyond just what we do on the clinical trial side of enrolling patients, gathering the data, the standards, really trying to take a thoughtful approach of how do we do what's best for the patient, have them make informed decisions, and get onto trials that potentially can impact, their life and the lives of their families, and get the data needed to impact, patients around the world are a key part. We also attend a lot of conferences, so it's always great to hear advances, new targets. And as that genetic data matures, as we understand the pathophysiology of how disease occurs, the mechanisms, we can develop new targets, new therapies to treat more and more of these patients. So I think that's a really important part. Something that we expect to see is more gene therapies for a broader range of, cardiomyopathies or heart failure or other kind of broad-based, large unmet medical needs, and, get patients away from the mentality of a transplant's the only option. And I think that's a key part, something that we work really hard to move forward.
Yeah, so you spoke a little bit about attending conferences, working with advocacy groups, working with experts that are both at the hospital or in the field. How does CTI stay current on industry trends and work with these advocacy groups?
Yeah, well, we're fortunate to be in an area with some leading academic centers, teams that are committed to treating rare disease patients with Cincinnati Children's and others. So I think inherently to CTI, there's a knowledge. Many of our coordinators, used to work at Cincinnati Children's or these other areas. They find their way to CTI and research and bring with them a lot of knowledge. Another key part of that is attending conferences. CTI is a sponsor and attends many rare disease focused conferences like World Orphan Drug Congress. We also attend ASGCT and Alliance for Regenerative Medicine's meeting on the Mesa. And that's a great opportunity to connect with investigators that have patients, understand where the gaps are in the treatment, to network with biotech or pharma companies with a novel idea, and to align ourselves with those advocacy partners to understand what that patient experience is as we think about designing a trial that is feasible. I was talking to an advocacy group just at a recent conference, and they talked about gene therapy trials specifically being, pretty labor intensive. We're giving something that has a high potential to impact their safety. We want to be really sure that we're monitoring this small group of patients before we expand it to others. And one thing they mentioned specifically for cardiac disease is that, these patients have lower cardiac output, they get fatigued, they get tired. We have a lot of assessments to do. And so how do you think about a schedule of assessments or a multi-day visit to break up that and make sure that the tests that they're performing are truly reflective of their disease condition, and it's also, not overwhelming to the patient that they want to feel like they have to drop out or they can't juggle work or their family lifestyle. And so I think we take a very individualized approach, to design these trials, but importantly at these conferences, meeting with key members to understand what that looks like. What does the future of cardiac gene therapy look like? What are the advances in vectors, or what is the regulatory kind of precedent at the time? And so I always enjoy attending conferences and those sessions to hear directly from the FDA at a working panel of what are the challenges, where can we improve, how to align ourselves with, current thinking on breakthrough designation or some of these other key components, and take that back to our teams, kind of collaborate on a strategy and to think through that with our partners. So for anyone attending any of these conferences, we would love to connect, hear what you're passionate about, what you're working on, how we can maybe impact or help support that mission as you go forward, whether it's, starting a Facebook group to try to spread awareness to patients, all the way to developing a novel gene therapy, across the globe. And so that's a great opportunity for our team, and would always welcome the opportunity to speak to others there.
Yeah, that's great. We can tell that, you're a client manager now, but you're able to draw on your project management experience and background to kind of give that full picture amongst the CTI project team to advance these projects forward. So,
Absolutely.
great.
Thank you, Bret, for all that you do. Thank you for giving us some time today, and thank you for helping to continue to move medicine forward.
It's been my pleasure.
Thank you, Bret.
Up next, Chain of Custody with Michael Schmidt, Clinical Project Manager 2, and Nicole Sparshott, Clinical Trial Manager.
So welcome to the podcast today, Michael and Nicole. Tell us about yourselves and about your role. Let's start with you, Michael.
Yeah, certainly. So prior to coming to CTI, I was at Ohio State, got a degree in biomedical engineering. And at my time at Ohio State, I worked in a benchtop lab and did research on nanomaterials, did a little bit of publishing work for that. And I think that was really at the point where I kind of got interested in cell therapy type science. So we worked a lot in nanomaterials. I kind of found my way into CTI by accident. I moved into their research associate program back in 2018. And from there, really moved into the ranks of project management, began as a project coordinator, and now I'm a clinical project manager. I've been mostly focused in the oncology-based research, specifically with cellular therapies. So certainly have gained a good breadth of experience with solid tumors, specifically in the cell therapy area. And I have found that I've also become pretty well versed in the chain of custody aspects as well. I've supported a few programs specifically in that arena. So certainly very excited to talk along that thread today with Nicole and the team here.
Great. Welcome. How about you, Nicole?
Sure. So I'm Nicole Sparshott. By trade, I'm actually a radiation therapist. So I've been in the industry for over 12 years, started off as a radiation therapist, was introduced to a clinical trial as a radiation therapy student, and it just piqued my interest. And then from there, I actually became an oncology clinical research coordinator many years ago. And then I transitioned to CTI here about 8 years ago and started as a CRA. Worked my way up, as Michael said, and then ultimately became a CTM at CTI in 2021, and was a part of the pioneering for that role here at CTI. So similar to Michael, all of my experience has been in oncology. I've worked on CAR T trials, cell therapy trials. I've worked in solid tumors, liquid tumors. Very familiar with the chain of custody aspect and all of the adverse events and safety items that you need to review as a CRA, such as like ICANS and CRS.
Yeah, great. Great to have you. So as you guys both mentioned, we're going to be talking about cell and gene therapy and more specifically chain of custody. So can you go into explain more about chain of custody, what that means in the context of cell and gene therapy trials, and then maybe why that is so important to the trials?
Yeah, certainly. So I think when we're dealing with cell and gene therapy trials, we're typically looking at indications of rare disease or very serious disease with oncology, for example. So not only is the patient very fragile in their state, but the product is also very fragile too. And what we see across different programs is there's varying ways in how that product is manufactured. So we take chain of custody very seriously, and at the onset of the trial, it's important to really get all the key players on the same lines of communication and understand what documentation, what steps, what escalations are all a part of that as the product makes its way through each of these steps in the chain, going from leukapheresis all the way to IP infusion in an autologous program, or if you're working in an allogeneic program, going from the cell bank to the patient. So really, we like to set up IP logistics plans. We work closely with sponsors in the development of pharmacy and IP manuals a lot of times. We want to make sure that these communication pathways are set up to a full extent. What's mostly important about the chain of custody is ensuring that we have matching of the patient identifiers. At the end of the day, we want to make sure that those subjects are getting the IP that is designated for them. That's all matched up through this full chain of custody process, where as the product moves along through the manufacturing cycle, we're ensuring that there's double verification of those identifiers at the site. We're reviewing that documentation in real time, usually through a validated platform where source can be shared in a private manner that is, compliant with regulations. And then most importantly, we're looking at whether there are deviations along this whole process. So you can have anything from temperature excursions to errors in the manufacturing process that can ultimately render that IP useless at the end of the day, or unable to be infused because the deviation is so severe and significant.
So what are some unique challenges that arise with cell and gene therapy as opposed to other trials, Nicole?
Sure. So there are a few different aspects to look at this. So we have limited patient population. So oftentimes, a lot of these cell and gene therapy trials are targeting rare diseases or specific genetic mutations. And so that really allows us to only pull from like a small patient population for these clinical trials. And so what CTI does is we really leverage our networks and our experiences with identifying and recruiting these suitable patients. We have a ton of collaborations. We go to a lot of conferences. We work with patient advocacy groups to help identify those patients for our sponsors. So that is one of the benefits of working with CTI, since we have all of that history and knowledge behind us. So we really capitalize on that in order to help our sponsors, but also for our long-term follow-up. So a lot of cell and gene therapies may have long-lasting effects. And so we need to consistently be reviewing the extended follow-up periods to assess for like the safety and efficacy of our patients. And so oftentimes there'll be a 15-year follow-up visit. So CTI has worked with our sponsors to help design and implement long-term follow-up plans, including patient monitoring and data collection strategies to track over time. So that is another thing that we work closely with our sponsors. And in fact, if they have multiple protocols we've suggested in the past and we've implemented, that they actually, all of those clinical trials can go into this one long-term follow-up. So we have worked with clients to do that as well. And then regulatory agencies are constantly evolving, and the guidelines for cell and gene therapy trials are, again, evolving constantly. And so CTI does have that regulatory expertise, so we can help navigate this complex landscape with our clients to ensure that we're compliant and with all of the regulations based on authority documentation that has been released. And then in addition, this whole process is rather complex. And so I think the benefit of working with CTI again is that we have that experience. It occurs at times where there's a ton of logistical issues that arise in these trials. And so we have had to pivot and implement, different strategies of how to best handle situations like that. For example, if a manufacturing company is on the West Coast and we are manufacturing a product on the East Coast, we've had to, at times, because of flight regulations, we've actually had to deploy an individual person to take that product over to the manufacturing company. So we really are thinking outside of the box, and we have that experience. So I think some of those things are what really identifies CTI as a strong leader with some of the challenges that arise.
From the project management side of things, and then also, you mentioned the CRAs, how do these different groups within CTI partner with the sites to ensure that they're trained and ready to receive the IP and, ultimately get to that first patient dosed?
Yeah, so I think as we begin to plan around SIVs for sites, certainly in feasibility in our pre-study visits, we're evaluating the suitability of their cell therapy laboratories as well as their apheresis units. So for the cell therapy laboratories, we ideally will go with sites that have FACT accreditations or JC accreditations, just so that they're compliant with those industry standards. And from an apheresis unit standpoint, we want to make sure that they have staff available and they'll be able to accommodate the specific nuances of the manufacturing schedule with the sponsors.
That's great. I mean, it's that hands-on opportunity to be able to work with the site and really make sure everything's set in stone before you're bringing in the actual product, the actual patients, and just making sure everyone's comfortable with that process.
So as Michael indicated, the CRAs are really helping and support this dry run process, and they're very involved with the project managers and the sponsors with whomever conducts it. Obviously, the CRAs are conducting the actual SIV where we'll go over, the slides of the IP manuals. The CRAs are very familiar with it. But in addition to that, as Michael talked us through, the lengthy process that's involved with chain of custody, the CRAs are then going on site and remotely reviewing all of these traceability documents. And from there, we're able to ensure that everything has been appropriately documented, that the patient has in fact been infused during the duration that is, written out per the protocol. And so I think it really just goes hand in hand. I think it's so important that we have that time with the sites at the SIV to go through all of these aspects to ensure that everyone's very clear on their roles and responsibilities. And then again, the CRAs are that second checkpoint. They're that point of contact for the sites for questions that arise. And then one other thing that is important is as we are going through these chain of custody and slot reservations, oftentimes with our phase 1 trials, these are competitive landscaping. So it's whoever is available first to take that first slot. And so it's important that the CRAs and the project management team are transparent with all of the sites with when the patient's product can be manufactured. And so the reason I say that is a lot of our phase 1 trials will have like a 28-day stagger between the first patient being dosed and the second. So the CRAs are really an integral part of that and ensuring that sites are aware, like, is the patient stable enough to wait almost 2 months from the time of screening, and then they go through their apheresis to when IP will actually be infused. So that constant communication with everyone is so important, and providing real life updates to the sites, letting them know where we are in the manufacturing process is another key component of the CRAs working with the teams.
Thank you, Michael and Nicole, for joining us today and walking through how the chain of custody works and how complex it can be and how CTI can help you. And we're so lucky to have experts like you helping us manage these trials here at CTI. And thank you for all that you do and for helping to move medicine forward.
Thank you.
Absolutely. It was a pleasure to be here today, and thank you all.
Thank you so much.
Thank you.
Up next, feasibility manager Joana Lancastre from Portugal. Welcome to the podcast, Joana. Let's get right into it. Tell us about the feasibility process for cell and gene therapy.
Feasibility process for gene and cell therapy studies follows the same steps as any other feasibility. But the big difference is that we are going to add a greater degree of complexity because we need to assess other capabilities in terms of staff and sites.
So how do you handle this complexity?
So I think when we start doing feasibility, the first steps, we need to carefully analyze the study protocol that we are going to implement and trying to answer some questions. Where are the patients located? What is the competitive landscape? What are the country requirements and experience? And what is the staff and site experience and capabilities?
How do you determine the best regional location for a cell and gene therapy trial?
So the disease prevalence and incidence is going to tell us the geographic location of the patient. And this is going to determine the country and site selection and strategy. And then we also need to do a competitive landscape analysis because the number and type of clinical trials that are going on right now in each country can influence the possibility and also readiness of sites and patients to join our clinical trial.
What is the process for picking the best site for a cell and gene therapy trial?
In terms of site profile, they all should include regulatory requirements, all the criterias in terms of good clinical practice for investigators and sites. And one of the key systems that we use here at CTI that we have in our department is our feasibility database. This is a system that allows us to keep track of global sites, investigators, and their disease expertise. We update very regularly the database, and it is always our first source to identify sites and PIs. And in this database, we include this information about their capabilities in terms of cell and gene therapy.
So you have a region and a site. Now what?
And then, so once the country and the final list of sites are approved by the sponsor, we reach to a second step of the feasibility, which is to contact the PIs and determine their interest in the study. So at this stage, it is here that we draft a survey, which will cover all the protocol specifics, the site's capabilities, and the PI experience. And for gene and cell therapy clinical trials, it is very important that we analyze certain criteria. For example, in terms of site staff experience, we want to understand if the investigator and the site have experience not only in the disease, but also in the conduction of complex clinical trials. Because these clinical trials, they usually have many visits, they have several assessments per visit, and they have very complex procedures. So I think to answer your question and to conclude, the site identification is not only looking for a site with a determined city or country. We also need to assess the PI relationships, their experience, the infrastructure, capabilities to conduct these very highly complex studies.
Thank you so much, Joana, for joining us on the podcast today. Up next, we discuss KRAS with Jaclyn Guillory, Director of Clinical Project Management at CTI.
Welcome to the podcast, Jaclyn. How are you doing today?
I'm doing great.
To start things off, tell us how you got into clinical research and how you came to CTI.
Yeah, absolutely. I graduated from the fabulous University of North Carolina at Chapel Hill with a degree in public health and kind of moved through industry, studying autoimmune diseases, getting into oncology, and finding my way here to CTI about 6 years ago. So we know that CTI has been a pioneer in the field of targeted therapies, especially concerning KRAS mutations. So Jaclyn, could you tell us a little bit about the significance of KRAS in cancer treatment and kind of how it, why it plays an important role? Absolutely. KRAS is really important. It's a gene that, when mutated, it drives the growth of tumors in various types of cancer, lung, colorectal, and pancreatic cancers. So when we're targeting KRAS mutations, it allows us to develop therapies that attack specific cancer cells. And so that minimizes harm to our normal cells and can potentially improve treatment outcomes for our patients.
A crucial component of developing these therapies involves identifying the right patients through genotyping. Can you explain genotyping process and the importance behind it?
Genotyping is a complex process. We're analyzing their DNA sequence for these, KRAS that we're talking about. It's really crucial because it helps us find the right patients with the right KRAS mutations. And that allows us to administer therapy to those who are most likely to benefit. It optimizes our efficacy and our safety of the treatment. So alongside genotyping, there's also HLA matching. Can you explain what HLA is? How does CTI approach HLA matching in the context of targeted therapies?
Great question. Actually, another layer of precision in our patient selection for our trials. So it involves matching the patients and the therapies based on their human leukocyte antigen or their HLA types. And these are proteins on the surfaces of cells, and they play a critical role in the immune system's ability to recognize foreign substances. So when we consider HLA types, we can further refine our selection for therapies. So again, our goal, making sure we are finding the right patients and optimizing their treatment.
Could you discuss how CTI identifies patients for trials with specific protocol requirements related to genotyping and HLA typing?
This is a really collaborative process between CTI, our sponsor, and our sites. We want to make sure we identify the right patients. A lot of the typing is done by the site. Some of that's done quickly. Some of that's done and outsourced, and it takes a little bit more time. Really coordinated effort on all parties' behalf to make sure that we get the testing done, that we're moving through to the process of manufacturing, and we keep the patient's best interest in mind. These are sick patients, so we need to make sure that everything is very well coordinated so that we can produce the therapy, deliver it in a timely manner, and maintain the integrity of the trial and optimizing everything for the patient.
So you mentioned manufacturing right there. What is like finding a slot for manufacturing? How does that affect the process?
Well, there are limited slots. So like a lot of things going on in industry right now, it's a really kind of juggling act between patient availability, patient's health at the time, moving quickly. So it's a really coordinated effort and requires close collaboration with our partners in manufacturing, a lot of good communication to make sure that we're well aligned moving into manufacturing and subsequently dosing.
Great. Do you have any final thoughts or messages for our pharmaceutical sponsors interested in exploring business opportunities with CTI?
Yeah, cell therapy, especially with KRAS, is really growing. I think there's so much potential, so it's really exciting. I'd like to say, CTI is dedicated to advancing the field of targeted therapy. We have expertise in genotyping, HLA matching, and patient-centric clinical trials. And we're committed, which, really position us to drive forward the development of these life-saving treatments. So we're excited about the possibilities of partnering with our clients to transform the future of cancer treatment.
Thank you. Thank you for joining us today.
Absolutely. Thank you for having me.
Thank you to all of our guests for giving us your time, lending us your expertise, and helping to move medicine forward. And thank you to our listeners for tuning in. Please make sure to like, subscribe, and share it with somebody that you think will like it. If you would like to find out more about CTI, please visit ctifacts.com.